All right, so we are about to record. No, there we go. All right, so this is Chapter 7, The Control of Microbial Growth. There is another 25-minute video that I think I've given summer 2015 students a link to, and this is um, the addition to that because that recording isn't complete, and so I won't be having a class between now and the time when I would expect you guys to watch that video. So this is me sitting in my apartment recording Chapter 7. Okay, so and I will try to. I don't think I'm going to be able to record as many jokes, actually, truthfully, but on we go. So, which of the following is not a direct method to measure microbial growth? And hopefully you realize the answer is 4D, metabolic activity, which is effectively how fast microbes eat. And the more microbes you have, the more you would expect them to eat, suggesting that you could measure the rate at which they eat, or the rate at which they consume oxygen, or the rate at which they evolve some waste product would correlate to the number of bacteria you have. And it would, but that is not directly counting the microbes as the other methods do. All right, so introduction, standards of sterility, microbial death rates, mechanisms of microbial killing. These are what we're going to cover in this lecture, and I'll touch on them briefly. Okay, so as far as an introduction, we already know that microbes are found in all Earth environments. Blood falls in Antarctica made by psychrophilic bacteria, as we saw at the beginning of Chapter 6 lecture. We've seen many times the prismatic spring, Grand Prismatic Spring at Yellowstone Park, and around the edge of that we can see the various different colors showing the different kinds of microbes that are thriving at various temperatures. Right? So we find microbes in all Earth environments. They are diverse in their metabolic capabilities, and we want them to die. These are things that cause human disease. Of course, as you guys know, most microbes don't necessarily cause human disease, but many do. And the fact that they do makes us want to kill them. Some microbes are harder to kill than others. This is a figure from your textbook that gives you a hierarchy of the most resistant to, and I think this is temperature, but you better not quote me on that, to kill microbes with temperature or to kill microbes with, yeah, and things like prions are very difficult to get rid of using temperature, and other things are significantly easier. And on this list are some things like prions and viruses with lipid envelopes that you might not have heard about. And I expound on that previous video. And this is not something that's critical, so I'll try to be very brief about it. Viruses are usually a capsid, a protein box, and that's shown in blue in this picture, and a piece of genetic material, usually, not always, but usually DNA, and that's shown in yellow in this picture. But some viruses, beyond having the basics, the protein box encoded by the genetic material, also have a phospholipid bilayer. And that phospholipid bilayer is pinched off of the host cell as the virus busts out of that cell. And as it leaves, it takes a pinch of the host cell's membrane along with it. The little spikes that you can see, these little spikes here and here and here, these are and this is HIV, by the way, this virus is HIV. Those spikes are proteins that are encoded by HIV's genome and they help these proteins bind to surface receptors on a human cell and allow the virus to get into that cell and spread itself from one cell to the next. Prions are misfolded proteins, and so these are proteins that naturally occur in your body or in the bodies of other mammals, and those proteins have a good way to be folded and a bad way to be folded. And you can see from the picture, as the prion bumps into the non-misfolded other protein, it's the same protein, two different ways to fold it. One's good, that's the normal protein. One's bad, that's the prion. When the prion bumps into the normal protein, it changes the shape of the normal protein into the prion shape. And you can see many prions, they sort of stick together and polymerize into a perverse quaternary structure for that particular protein. And this diagram is pretty accurate because these are sharp and pointy, and many of these are made into sort of a sharp and pointy object that can poke holes in cells and make cells die and cause disease, such as scrapie or mad cow disease. Both of these show the normal shape of the protein on the left and the prion protein on the right, and we have secondary structure, alpha helices here, beta sheets here, and you can see these two surfaces of beta sheets would allow you to have one surface stick to the back, another surface stick to the front, and therefore having the beta sheets allows it to polymerize. And again, this you better not ask me how this protein can get from your stomach into your bloodstream and into your brain, but apparently it does. And if you eat it, then you can get mad cow disease. But mad cow disease is extremely rare because mad cows were all destroyed, and they're no longer fed in the way that might cause them to get mad cow disease. And of course, the way that is them feeding on them. Yikes. Anyway, can't believe they actually did that, but that's the way it was. Yeah. Okay, so on to subject matter, effectiveness of treatment. If we want microbes to die, no matter what they are, no matter how hard they are to kill, if we have a killing treatment, and whether that killing treatment is high heat or radiation or a chemical disinfectant or whatever it is, how the microbes die is going to be dependent on some variables. The number of microbes, in other words, the initial population, as you might imagine. The greater the initial population, the harder it will be to kill all of them. And the longer it will take for all of them to die, however many you have. Likewise, the environment. If there's a biofilm, microbes in a biofilm are harder to kill. So the environment, the temperature, the organic matter, etc., definitely matters to getting microbes to die. The time, again, the longer you expose a microbe to a lethal agent, the greater percentage of the microbes in your population will die. The concentration of the killing agent, whether that's radiation, a lot of radiation kills more than a little if that radiation is lethal to that particular microbe. The type of killing agent, well, different types of killing agents kill in different ways, and different types of killing agents will be more or less effective on different microbes. Likewise, the mechanism of action can vary from treatment to treatment from treatment. There are many things to target with killing agents, such as DNA. You destroy DNA, the cell can't make proteins. And if the cell can't make proteins, it dies. You destroy the proteins, well, then the cell dies, as previously mentioned. You destroy the membrane, well, then the cell falls apart and dies, etc. So there are lots of targets for chemicals or radiation or other killing agents to work on, and each particular killing agent has a mechanism of action by which it kills. Microbial characteristics. So, gram-positive cells, gram-negative cells, cells with a glycocalyx, cells that are able to form endospores. Those variabilities within the kinds of microbes you're trying to kill will have an effect on how effective your treatment is in killing them. Okay, so it is very difficult to obtain sterility, so I often talk during lecture extensively about this slide because I like talking about Mars and going to Mars and so on. When we send space probes out to other planets, well, should we be concerned if those space probes are not sterile? Well, if they're going to land in places where we think there might be life, I would submit to you that it is a concern for many reasons. So one reason is that if we were curious as to whether there was life or not, well, if we found a contaminant, we might be confused about whether that contaminant was a exobiological element, in other words, a piece of life from another planet. In other words, life on Mars if we went to Mars. And you can see I didn't capitalize Mars at every instance, and my apologies for that. Yeah, so getting a false positive. Woohoo, there's life on Mars! And it looks just like Bob's microbes that were in his nose. Wow. You can really get around, Bob. Anyway, so you can see the problem with that. Hopefully that problem might be traceable, but maybe it wouldn't be so easy to know for sure if that was a microbe that was coincidentally similar to a human or Earth microbe, or whether it was a contaminant. Secondly, if there was life on Mars, well, then we just contaminated Mars, and as an answer to the question, would there be competition between Earth microbes and Mars microbes? The answer to that is clearly yes. Evolution tells us that when you put a living thing in a particular environment, that living thing will try to survive in that environment. And if it's successful, it will proliferate. And if it proliferates, it's not going to care whether or not whether it doesn't belong there or not. Yeah. So this idea of invasive species and diseases going to Mars is not a new one, and I think I put it in a slide a couple minutes ago that talks about that. And so the idea of which would win, well, you might expect a home field advantage to make sure that Mars microbes outcompeted Earth microbes, but that's not always the case. Earth is filled with examples of what are called invasive species that win their away games, so to speak. Okay, so there's one other point that I think is worth discussing, and that is that if one of these guys, and you can see that these guys working on this space probe are wearing special equipment not to keep the space probe from infecting them, but to keep them from infecting the space probe. They're wearing clean suits because they don't want to contaminate the space probe. And again, if they do contaminate it, getting that space probe clean is going to be very, very difficult, right? Space probes cost $1 billion, and if they cost $1 billion, then you're not going to want to treat them with treatments that obtain sterility. So the bullet point I have here is treatments to obtain sterility are often destructive to the thing we're trying to sterilize. When we're in lab, we sterilize things with open flame. We're not going to put $1 billion space probe in the open flame because it will melt and then it won't go to Mars very well.